Okay, the male and female reproductive system. So we're going to talk about the gonads, like I began to say. So the gonads are important, and you'll know all the parts, gross anatomy, and a little microscopic anatomy, a little histology as well. So the male has the testes, and the female has the ovaries, and there's two of them on each, two ovaries, two testes. What you're going to see about the male gonads is these are outside the body. Ovaries are deep inside the body, in the pelvic cavity, peritoneum. So the reason the testes are outside the body, because they need to be, I would say, about 3 degrees below body temperature. So if body temperature is 37 degrees Celsius, the testes need to be 3 degrees less. Now, why is that? Because they have to make the sex cells, which are called gametes. There's gametes in both. So the gametes, sex cells, are the testes have the sperm, and the ovaries have the oocytes. Pretty straightforward, right? Sperm is very easy to make. It's made pretty much your whole lifetime. Oocytes, not so simple. Oocytes, you make millions of these things before you're even born, but you really only get, I would say, 400 to 500 viable oocytes to be fertilized by the sperm. So again, reproduction is not so much about um, sexual activity, right? It's about getting your genes, your chromosomes from one generation to the next to keep perpetuating the human being, right? So that's really the whole idea. And the diversity of that is very important because you're going to undergo not mitosis. Sex cells, gametes, don't undergo mitosis. I mean, they do it first. You know, the stem cells undergo mitosis. But in order for sperm and oocytes to divide their nucleus, they undergo what's called meiosis. That's sex cell nuclear division. So basically, you're going to, after meiosis, and there's two phases of meiosis. There's meiosis 1 and meiosis 2. And, you know, this stages of all PMAT, right? Prophase, metaphase, anaphase, telophase. So that's all one, 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 one. And then there's PMAT 2. So that's really important. The sperm, though, straightforward. You get one, and you're going to hear terms like, oh, a spermatogonia, which is gonia is a very immature stem cell, really. And that technically undergoes mitosis. So they start with mitosis. And the oocyte stem cell is called oogonia. But ultimately, after 2 meioses for the male, you're going to get 4 completely different haploid cells. Haploid means they have 23 chromosomes, not 23 pairs. That's 22 autosomes, one sex chromosome. And they're completely different after meiosis. Mitosis, you get identical cells, like through somatic cells, like epithelial cells. Now, female, of course, is much more complicated. Females, you begin meiosis 1 before you're even born. And meiosis 1 doesn't complete. It's first of all, it stalls in prophase 1 in the ovary, the oocyte, which starts with an oogonia, then undergoes mitosis to become a primary oocyte. And then it stalls in prophase and completes meiosis 1 when you reach puberty. Okay? So then it stalls again. It starts meiosis 2 and then it stalls in metaphase 2. This is the ovary. Now this is the oocyte. Now this stalls in metaphase 2, and it's complete only with fertilization. So you'll see what happens in lecture. We're not going to talk too much about meiosis, but you have to understand this whole part before we can talk about how the reproductive system looks and how it works. So in the female, the oocyte will be stalled in meiosis 1 until puberty. And you know what happens at puberty, right? You get a release of GnRH, gonadotropin releasing hormone, from the hypothalamus, which stimulates the anterior pituitary to release FSH, follicle stimulating hormone, and luteinizing hormone, LH. So that's really important right there. And then you start puberty, and then you have a surge, of course, of estrogen because of the FSH release. I'm sorry, because of the LH release. LH is what stimulates the granulosa cells to produce estrogen. So estrogen is the mother of all the hormones in the female reproductive cycle. And then meiosis 2 will begin and will stall in metaphase 2. And once that sperm, you know the story, right? On a Friday night, the sperm will swim through the uterus and then go into the uterine tube and then meet up with an oocyte that was ovulated on the 14th day of the uterine cycle or the ovarian cycle. And then the sperm will dig into the oocyte, and those nuclei will be in the same cell, and you'll get what's called an ovum, and that ovum now completes meiosis 2 and gives off another polar body, which you'll learn about. But really, the only cell that's a viable oocyte is called the mature secondary oocyte. You'll see the polar bodies are not fertilizable, if you will. So you get one of these a month, basically. So, and it seems like the ovaries will alternate from right to left. There's 2 ovaries each month. And on the 14th day of your menstrual cycle or your ovarian cycle, based on a 28-day cycle, you'll ovulate, you'll release a secondary oocyte from the ovary, and that will make its way to the uterine tube, and it'll meet up on a Friday night. This only happens on a Friday night, the 14th day. You'll have a fertilization, and the fertilization should take place in the uterine tube, which is the fallopian tube. Okay, and then I'll talk a little bit more about what happens after that as you learn the actual parts of the reproductive system. So I'm not going to ask you this, but there's going to be a picture of an ovary, and the ovary will have oogonia in it, and you're going to have to learn about a follicle too, because like if this is a follicle in your ovary, the oocyte is inside. It looks like a cell, right? So the follicle is like a spaceship that kind of keeps the oocyte nice and cozy and feeds it, and then all these cells are producing estrogen. So you have to see an oogonia is going to be inside a follicle, and the primary oocyte is going to be inside a follicle, secondary oocyte, right? And then the definitive is like the most mature. I don't really like that term. I like to use the term Graafian follicle. It's the maturest follicle. Seems like the ovary picks the, I'm sorry, that's the follicle. That's the follicle, but the oocyte is your secondary oocyte, which is the mature oocyte, but it's in a Graafian follicle. So the ovary seems to pick the most mature of the oocytes to be ovulated, and the other ones kind of wait around till next month. So maybe they'll get picked next. You know how it goes. So this table will probably help you more for a lecture than it will for lab. It's very good, actually. So here's the ovary, and this is the cartoon on top. So it's showing follicles. So you have your primordial follicles, which contain oogonia. Oogonia are the immature cells. When you hear gonia at the end, that's immature. Stem cells, really. Stem cells. And they have to undergo mitosis first because they're 2N, they're diploid. But then you have your primary follicles, which contain basically primary oocytes, right? Primary oocytes. Now, the primary oocytes will undergo meiosis. They'll start meiosis anyway, and they'll, of course, before puberty, it'll stall in prophase 1. But by puberty, now you got this secondary follicles, and you have a secondary oocyte. I'm going to say it down here, though. Here's the secondary oocyte, because technically this could still be a primary oocyte. That technically it can be, but I wouldn't split that hair. If I point to this, you're just going to say primary oocyte or secondary oocyte would be fine. So now we have a nice mature follicle with a mature secondary oocyte. So it's still stalled in metaphase 2, and the zona pellucida is the outside of the oocyte. So zona pellucida is what the sperm has to penetrate to get into the oocyte. Zona pellucida. It's a glycoprotein covering that the sperm kind of digs into. Then you have, on the 14th day, that Graafian follicle, I'm going to call that a Graafian follicle. This Graafian follicle, this is the mature follicle, will rupture and expel and ovulate the secondary oocyte. And it doesn't go anywhere except into the peritoneal cavity. And you'll see how the uterine tube is going to snatch that up and bring it into it. So what happens after that, this is really important. You have a little bit of an in-between, and the corpus luteum is the next phase. The corpus luteum is really important. So after the 14th day, after ovulation, the ruptured follicle, which sometimes they call it the corpus hemorrhagicum because it bleeds a little bit. But the Graafian follicle that expelled the oocyte is going to become the corpus luteum. And that's going to hang around for like 14 days after the ovulation, because this is releasing hormones. The corpus luteum is releasing hormone. Remember, you have like LH, which is luteinizing hormone. It's also released in the male, though, you should remember. So luteinizing hormone targets the corpus luteum to release estrogen and also progesterone now. Now progesterone comes into the picture. So the corpus luteum is secreting. So you have a luteal phase of the ovarian cycle, and you have the secretory phase of the menstrual cycle. So the corpus luteum secretes estrogen, which is important for all functions of the female reproduction, and progesterone. Progesterone is a steroid hormone that will now begin to prepare the uterus for implantation. Just in case a sperm, again on a Friday night, the sperm swims up and penetrates that zona pellucida to fertilize that egg in the uterine tube. So progesterone prepares the uterus. Now, let's say it didn't work out this Friday night. We didn't meet a nice sperm. The oocyte did not hook up with a nice sperm. Then the corpus luteum now has really no reason to hang around because there's no implantation in the uterus. So the corpus luteum basically becomes the corpus albicans and undergoes what's called atresia, which is degeneration, really. And that's what happens to all the oocytes that aren't fertilized. They just undergo atresia. And hopefully the next mature oocyte that was waiting in the wings will come out next month and have another chance for a nice sperm to come in. So the corpus albicans only shows up in an unfertilized uterus. So, but the corpus albicans is what the corpus luteum becomes if there is no implantation. And you'll go through APR and you'll see the histology. And you should know what a sperm looks like. It's very simple, the sperm. Again, the male reproductive system is nuts and bolts, very quick, no pun intended, to fertilize an egg. It swims, it has a tail, and millions of these things are released, but really only 200 will make it to this point. So it's really survival of the fittest for the sperm to get from the testes all the way to this uterine tube. That's a real big, they'll get a lot of credit card miles on their cards. And it's like a Tough Mudder. They really have to get there. So here's some histology, which again, you'll see this. You have your assignments through APR, dissection. Of course, the female is always a little more difficult. So again, showing the primordial follicles, which are 2N. They're diploid. I shouldn't say that the follicles are diploid. The primordial or oogonia is 2N. And then here's your primary follicle. And this is your mature, or I like to call it the Graafian. I just like saying Graafian follicle. And that contains a haploid, just an N secondary oocyte. Because it went through meiosis 1. Because now we're in, you have to be going through puberty to get this one. Otherwise, they're just kind of waiting for you to go through puberty. So here's the oocyte. You have your primary oocyte, which the oogonia will become after mitosis. And then you'll go through meiosis 1 and after primary oocyte, and you'll get another primary oocyte, but that will become a secondary oocyte. So this is kind of a gray area here. So if I point to this on the test, you're just going to say oocyte and you'll be fine. Right? You'll be fine. But the follicles are more important. You should know whether, like in the picture before, you should know that these are primordial follicles and these are primary follicles here. And this is your secondary follicle. And this is the vesicular is fine, or Graafian follicle. This is ovulation. This is the corpus luteum. This is the corpus albicans. Keep it as simple as that. And if you look at your list, your checklist, that's the things that I need you to know. And it's pretty inclusive. So this is histology of the Graafian follicle. Again, I'm going to go by APR. I'm not taking you off of these pictures, but it's exactly the same. Corpus luteum, and here's your corpus albicans. So it's a little memorization. You have to, find these things in APR and check them off with your list, and you'll do really, really well. I'm not going to get this detailed on my exam, and I'm sure it's not going to happen on lecture either. So don't worry about getting too close. We will find out what the ovarian cortex is when we look at the APR, but just to show you that. So you notice, remember what cortex is? Cortex is outer, right? So that's all this. This is the cortex of the ovarian cortex. It's not clearly defined. And this in here is all the medulla. So it looks like all the fun stuff is happening in the cortex. Don't you agree? Like in the cortex is where you have all the follicles with the oocytes. The medulla is just blood vessels and lymph and nerve tissue. So just keep that in mind too. Because I will ask you the difference between the ovarian cortex and the ovarian medulla. It's a simple question. Here's a nice close-up though of the secondary follicle, or the mature follicle even. And you can see here is your secondary oocyte. And these are what's called the corona radiata, which is basically granulosa cells. It's the same thing as granulosa cells out here and here. I won't ask you about the theca externa or interna, but you might have to know those for lecture because they're slightly different in what they secrete. So granulosa cells are all about secreting estrogen. That is the serious mother of all female hormones. The zona pellucida is actually the outer membrane. You can call it a membrane, I guess, of the oocyte. So the zona pellucida is what the sperm has to penetrate to get in there because the nucleus is inside. And remember, it's in the secondary oocyte is haploid 23 chromosomes, right? It's not 46 chromosomes like our somatic cells because this is a gamete. Yeah, this is a cloudy area of granulosa cells. There's massive amounts of estrogen being released here. And some of the parts, and I forgot to mention this, is the antrum. This is a fluid-filled area within the follicle that, again, helps nourish the follicle. And you get massive amounts of estrogen being secreted in here as well. And other nutrients. There's glucose in there, too, and sodium, potassium, electrolytes. All that this little cell needs to survive to eventually get ovulated. So here's the parts. Here's a little bit of our parts. And again, just to notice, I probably won't ask you about the theca externa and the theca interna, but you got to know what an antrum is, corona radiata, granulosa cells, and the zona pellucida. Those are important. Now we're into what's called the uterine tube. So let's take a look. Again, we're going to go to APR later and look at the gross anatomy. So this is the uterus right here.
Sorry, before that, can I have a question about the oocyte?
Sure.
Can I also call oocyte as ova or ovum?
Yeah. Ova is plural for oocyte.
Okay.
Yeah, very good. Yeah, that'd be fine. That'd be fine. It means the same thing to me. It means egg, right? Very good. So here's the uterus, and this is the opening of the uterus, which is called the cervix. There's only one uterus, right? But there's two ovaries. Here's your ovaries. Not my ovaries, your ovaries. And then you have, and now the ovaries aren't even technically part of the uterus. This little structure that connects the ovary to the uterus is called the the ovarian ligament. That's important to know. And you'll have to identify that. Because the ovary is not real. This is not a tube. This is just a ligament right here. But the thing on top, it is connected through a tube to the uterus. It's called the uterine tube. Now, the uterine tube is also known as the oviduct. Makes sense, like the egg goes through this duct, exocrine. And it's also known as the fallopian tube. So the fallopian tube is connected to the uterus, and it does allow two things to happen. And I'll show you in a minute. So let's look at that. Now, on a Friday night, right? Friday night, the sperm, right? The sperm with the tail, the mitochondria is right here in the middle piece. Here's the head of the sperm. The sperm will swim up into the cervix, swim into the uterus, and then into the uterine tube, all the way to about here, hopefully. Now, the uterus is going to ovulate a secondary oocyte, 14th day, right? Hopefully on a Friday night. And then at the end of the uterine tube is a structure called fimbriae. Fimbriae is like a catcher's glove for the oocyte, secondary oocyte. It'll catch the oocyte because the oocyte just released, ovulate into the cavity. So it can be there for a few hours. So hopefully by midnight, the fimbriae will catch that oocyte and bring it down to this area to meet up with the sperm. And I'm just going to draw it on this side. This area of the fallopian tube, or the oviduct, or the uterine tube, it's all the same thing, is called the ampulla, which is like a little bit of a dilation of the tube. So that's the ampulla of the uterine tube. And that's where you want to have fertilization. You don't want to implant the ovum there, but you want to have fertilization there. So that's the site of fertilization. Site of So what that means is the sperm, the head of the sperm will penetrate the zona pellucida of this secondary oocyte. So basically what this is showing is the histology of the uterine tube. And it has a lumen, and it has cilia, right? Look at this epithelium. This might look familiar to you. It looks like a simple columnar epithelium with cilia, because the cilia is going to move the oocyte to where it's supposed to go, and that's the ampulla of the uterine tube. And there's muscle layers too. It doesn't show the muscle layers so good, but here's the muscle layer. And it's peristalsis. It actually contracts circularly to move, of course, involuntarily. Smooth muscle contraction to move that oocyte to where it's supposed to go. So this is the histology of the uterine tube. And we'll look at more. And here's the layers of the uterine tube. And ampulla, of course, is really important. Fertilization most commonly happens in the ampulla. So you're going to have to point that out on an actual model or real anatomy. Right? And I won't ask you about the isthmus or any of that really, but the layers you need to know in the histology. The mucosa is the inner layer. Then you have the muscularis, which is smooth muscle that helps for peristalsis. Serosa is always outside of an organ or a tube. Mucosa is always the inside. And there's cilia, right? Cilia. Now, not only that, but things happen, like you're going to learn this in lecture, like especially during ovulation on day 14, within 48 hours of day 14, the uterine tube and the uterus are welcoming the sperm. They're trying to help the sperm come in and get to that ampulla of the uterine tube. So Most of the month, though, it's not so easy because the mucus is thicker in the female reproductive system, the cervix especially. But during that 14-day, 48 hours during ovulation, the body changes in the female to facilitate the sperm. And also pH is important. Like the vaginal pH is usually pretty low, more acidic, except around the 14th day, and everything changes. And hopefully, again, that's a Friday night, so you can have fertilization. So this is the uterus, the layers of the uterus. And there's 3 layers of the uterus, which I'll show you more clearly on the APR. So here's the lumen. So remember on a Friday night, the sperm meets the egg in the oocyte in the ampulla of the uterine tube. So it takes about 6 days or so for that fertilized ovum, and it becomes a zygote because now the 2 haploid cells, nuclei, have fused, and now that's your first cell. That's you, right? That's going to be you. That's the first little Victoria. She starts as 1 cell zygote, which is diploid now, and that's going to differentiate into all of the layers of, or all the tissues. So it takes 6 days for that zygote, and it changes and it undergoes mitosis or cleavage till it reaches the uterus, again through peristalsis and ciliary movement, moving the ovum, the fertilized egg or the zygote, to the uterus. So that's going to be in the lumen of the uterus. Now the innermost layer of the uterus is called the endometrium. And this is different phases, it's showing you different phases, which I'm not going to ask you. But you do have to know the inner layer on a model or real anatomy is called the endometrium. And that has 2 layers, a basal layer, which is always there, and a functional layer that only shows up in the presence of progesterone. So this starts to build up, like maybe the 8th day of your menstrual cycle, and because of the release of progesterone. So this builds up until it gets really thick, right? And thick because it's expecting to nourish an implanted zygote, which is really blastocyst by then. And that's where you have pregnancy, right? If that zygote implants in the endometrium, then you're pregnant, and it's going to release a hormone called HCG, human chorionic gonadotropin, which the metabolites break down in the urine. So you can pee on a little stick, and it's checking for HCG, and it'll say pregnant or positive or whatever. Okay, so the functional layer is really a place that's temporary until there's an implantation. Now, if there's not an implantation, if unfortunately we didn't meet a nice sperm on a Friday night, then the functional layer is going to shed and be removed from the uterus. And that's the menstrual phase of the menstrual cycle, the first, let's say 7 days. Okay, and that's when progesterone drops and estrogen drops, and then you get sloughing off of the functional layer along with the arteries that have been feeding this tissue. So it's a little blood loss and tissue loss, and that's the menstrual flow. So know the 2 layers of the endometrium, the functional layer, which is temporary, and the basal layer, which is always there and always has blood supply. So here you go. And I didn't mention the outer layer because there's really not much to talk about with the outer layer, just called perimetrium of the uterus. Now, this is the uterus. So you get your list down, you go through the first 2 pages of the checklist, and then you have your word bank at the end. So this is the cervix, and that has epithelium and muscularis just as well. There's an internal and external opening, like it's called the internal os, actually, you'll see that. That's short for ostium. And then this is the cervix now. Remember, this is the cervix we're talking about here. And this is the histology. And your external os. Okay, and that's the epithelium. Here's the lumen. Always know this is the lumen. That's important. So you have epithelial tissue. Kind of looks like skin a little bit, doesn't it? And then you have your lamina propria, which is usually areolar connective tissue. And then you have the muscularis layer, which for peristalsis. and facilitating sperm sometimes. Right? So that's important. But there's also mucus being released from the mucosa. Now, the sperm has to fight through that mucus to get to where it's going on a Friday night, which is the uterine tube. So sometimes the mucus is very thick, especially outside of that ovulation period, that 14th day. But that's really ideal. So here's a little more straightforward. This is the male reproductive system, and here is the testes. And you'll see this on a gross anatomy, so you'll see what that is. And this structure that covers the testes is called the epididymis. So you can see, like, this is tunica albuginea, is basically a connective tissue that holds the testes in one place. So, and remember, there's two testes. They're outside the body in a covering called the scrotum. There's two on each side. It's outside the body. So again, in lecture, you're going to learn that within the testes, you have what's called seminiferous tubules. And seminiferous tubules are the site of spermatogenesis. And that's making sperm. And that's targeted by your FSH from your anterior pituitary. And the cells that do this in the seminiferous tubule are called sustentacular cells or Sertoli cells. So basically, they help nourish the sperm so they could develop. So Sertoli cells are important for spermatogenesis. So the FSH targets Sertoli cells, which are called sustentacular cells. Now, interstitial cells are between the Sertoli cells or between the tubules, and there's another name for them. They're called Leydig cells. And Leydig cells are targeted by LH, and Leydig cells produce androgens like testosterone. Just like the granulosa cells produced estrogen, the Leydig cells produce testosterone, which is the main sex hormone, which is endocrine into the blood, right? This is not going into the urethra. The sperm is going into the urethra. So the sperm goes through that meiosis 1 and 2 all the time after puberty, of course. And the spermatid is what you see after 2 meiosis. But then they're going to mature. And a mature sperm is called a spermatozoa. A very immature stem cell of sperm is spermatogonia. So spermatids already went through meiosis, so they're haploid. Spermatozoa is just almost ready to go, and they're haploid. Spermatogonia are diploid. So they have to undergo mitosis before they can start meiosis, which you'll see. And the tubules have a lumen, where the more mature cells will go into the tubular system of the seminiferous tubules. And then there's a place right here, like right before it goes into the epididymis, the sperm, and there's millions of these things. It's called the rete testes, which you'll see again on the APR. And then you have the epididymis, which is a storage place. This is the epididymis out here. You'll see it on the gross anatomy very easily. Like, this is the epididymis. And the epididymis is like where the sperm really mature and they learn how to swim. So they probably take swimming lessons in there. They lift some weights. So the epididymis is like a boarding school or gym where the sperm get ready to be able to swim and get to that uterine tube. So when the time comes, you know the story, on a Friday night, the sperm will begin its ejaculation process through a tube called the vas deferens. So you have to know that too. That's when you'll see a lot on the gross anatomy. And this is also known as the ductus deferens. So look at the different names. It's pretty common. There's probably a list coming up. This is a little higher, lower magnification of the tubules. And you can see the Sertoli cells. Interstitial cells are between. That's why they're called interstitial cells. And the lumen, which is over here, in this high magnification. So it's a tubular system within your testes, and it's a very long tubules. Like if you pulled these tubules out, you could probably climb to the second floor of WCC Science Building. but don't do that. Okay. So that's basically the flow, and I'll show you on the gross anatomy and when we get there. So here's another table. The immature spermatogonia, which undergo mitosis to give you another diploid cell. And then you start meiosis. The primary spermatocyte undergoes meiosis to become a secondary spermatocyte, and that will mature to a spermatid a little bit more. They differentiate a bit. You don't need another meiosis. That's done. And then the spermatid will mature to a spermatozoa, especially in the epididymis. Get ready for that Friday night. So spermatozoan is singular. And here's the two cells. And again, this is probably more for lecture to know the two different cells. And remember, LH, because L targets interstitial cells to produce testosterone, and FSH targets the Sertoli cells to aid and facilitate spermatogenesis, making sperm. So interstitial and LH is more endocrine, where FSH is more the exocrine, the sperm that's going to be ejaculated out the urethra. And one thing I forgot to tell you with the male and female, the male urethra is shared with the reproductive system and the urinary system, whereas the female, you have different orifices. You have the vagina and then a separate urethra. That's important, and you'll see that on the models, and you see it on the real anatomy too. So this is the epididymis. This is the gym. So it kind of looks like the seminiferous tubules because it's kind of an extension. So the lumen is where the mature guys, these are the guys who are ready to be deployed during ejaculation. Only on a Friday night, right? Is that true? Who knows? Jonathan, what do you say? Okay, so that will, again, has cilia. And now we have, this one has pseudostratified. You don't have to know that. You've already been through that. And there's a muscularis to it because it's got to be released through, there's two muscles. You're going to see the dartos muscle and the cremaster muscle will actually propel the sperm out. If you look at the list, like the two checklists, I'm looking at that right now, the male is almost half of the female. So there's less structures. And don't forget to do the sperm. It's very simple. If I don't get to that today, make sure you look at the sperm. It's on your list. It's very simple. Tail, mid-piece, head, period. So we're going to look at other things. You're going to see the epididymis on the gross anatomy. You'll see the vas deferens on the gross anatomy. And then we'll look at these three glands. The prostate gland, there's only one of those. There's two seminal vesicles, and there's two bulbourethral. So these three will add to the semen, the fluid that's in the ejaculate that facilitates the sperm getting to that fallopian tube, ultimately. So it has to go through an acidic urinary tract of the male. Then it has to go to a possibly acidic vagina. Then it has to go through thick mucus of the cervix. Then it's got to continue swimming up through the uterus into the fallopian tube to the ampulla. So the prostate gland is going to produce and secrete some citric acid, which is going to help the mitochondria of the muscle of the sperm to give it energy, to fuel for energy. So citric acid is like a version of vitamin C. And there's a PSA, you'll learn this in lecture, is an antigen that's on the prostate. So the prostate should have no PSA, because the more PSA that's on that prostate, the more chance that it has cancer or enlargement. Hypertrophy is enlargement. Hyperplasia is too much mitosis in the prostate, which could be prostate cancer. So this should be 0 with people your age. But as you get older, you can get away with like a 3 or 4. So PSA is used really to check either prostatic hypertrophy or prostate cancer. But really, it's a gland that secretes fluid into the semen, mostly citric acid. The seminal vesicles will produce and secrete something like fructose. Fructose also will give energy to the mitochondria of the sperm. And this is the most abundant, or the one that adds the most to the semen. Also, there's prostaglandins. Prostaglandin is a chemical that kind of breaks up the mucus and helps the sperm stay together. So they're friends in this tough mutter at the beginning, but then at the end, you could have 150 million sperm released during one ejaculation, but only 200 will make it to the ampulla. So this is survival of the fittest, thanks to these glands that are producing these facilitating fluid in the semen. Bulbar urethral gland is mostly about mucus, especially helping out on a Friday night with sexual intercourse. So lubrication. But all of them together, especially these two, produce alkaline fluid, which is base. So pH below 7 is acidic. So you want to make sure that the sperm can survive in that acidic environment. So these glands that producing the semen also have an alkalinism or alkaline fluid to neutralize the acid in the urinary tract of the male and female reproductive system, and also help the sperm survive. So you're going to have to point out the prostate gland, the seminal vesicles, and if we could find the bulbar urethral gland, which we did, I saw it on the APR. So those are really important. All these things on this page you're going to have to name on the exam. More histology of this is the vas deferens, very muscular, very muscular. Look how small the lumen is in relation to the tube itself. So the lumen is where the sperm is swimming. And of course, there's cilia and there's muscularis for peristaltic contraction to get that sperm out of the penis and ultimately out of the urethra penis and then into the uterine tube. So take note in the APR, the histology part that you're responsible for. This is the seminal vesicle, and this is the gland. Don't get this confused with the seminiferous tubules. Seminiferous tubules are in the testes, right? Those are the areas where sperm is made through spermatogenesis. So this is the seminal vesicle. This is the gland that produces most of the semen, and this is where the fructose and prostaglandins are released from into the semen. Here's the prostate gland histology. And the thing about the prostate is, if you notice the urethra, where the sperm is going to be expelled, goes right through the prostate. So this thing's enlarged. It's going to affect your urinary system. It's going to affect the release of urine because it kind of occludes the urethra. So we'll see where the urethra is when we do the APR, the more gross anatomy. This is the penis and a cross section. So look, this is the urethra down here, right? So the outer part of or around the urethra is a group or tissue. It's erectile tissue, which is very vascular, called the corpus spongiosum. Then you have these two other cavernous areas of tissue called the corpora cavernosum, which is also erectile tissue. And those you're going to have to point out on the cross section and the longitudinal section, the corpus spongiosum and the corpora cavernosa or cavernosum is one. Cavernosa is two. So there's two corpora cavernosums and there's one corpus spongiosum. That's erectile tissue. There's connective tissue in there. There's very little muscle in that tissue. So this really relies on blood flow, especially on a Friday night when you want to get that sperm to the uterine tube to have your DNA be transferred to the next generation. That's the only reason this all happens, right? Here's some things you need to know when you look at the gross anatomy. The broad ligament. Good luck with that one. It's just a ligament within the female reproductive system. The ovarian ligament I already showed you. All right, and I think that's pretty much it for the ligaments. Maybe the suspensory and the round ligament too. So you have to see those on the APR. So if they're on your list, which I think at least two of those are on your list, broad ligament is, ovarian ligament definitely, and you'll see the list. We pretty much hit everything except for the gross anatomy so far. So here's what we're going to look at later on. This is, again, it's not a model. This is just a drawing of the uterus and the ovary. So we already showed you the ovary. But now you got a nice close-up here. And you can see the cervix right here, which is the opening of the uterus. And you're going to have a fundus and a body of the uterus. You'll see that on your list, I believe, yes. And then you'll have the broad ligament, the ovarian ligament. Here's the fimbriae of the uterine tube. Here's the uterine tube. Here's the ampulla of the uterine tube. Here's the ovary. Here's the ovary. And this one is just open so you can see the follicles. Right, and then the ligament right here, the suspensory ligament. And here's the uterine cavity, I guess we call this the cavity, the internal os and the external os of the uterus, and same thing you have in the cervix. So you'll see that again on APR when you're doing your practice, or you're doing your study first, then you do the practice. And below this would be the vagina, right? The vagina would be below the cervix. So we'll look at all those things in APR. This is the best way to do it, because I'm not going to give you this picture. And notice the female reproductive, and this, you're going to see a lot of this. This is like the mid-sagittal section or the lateral view. And you can see now we'll look at things like the urinary bladder. The uterus is on top of the urinary bladder. So you can imagine in a pregnant uterus how much pressure the urinary bladder has on it, because the uterus is on top of the bladder. So you have to know that this is the vagina here, and this is the urethra, and this is the anal canal right there. So there's 3 openings there. And we'll go through the external anatomy, like the mons pubis and the labia majora and the labia minora, the vestibule, the clitoris. And then again, you can see the fimbriae and the ovary and the uterine tube here as well. So remember, the reproductive organs of the female are inside the cavity. And we'll go through these. I have to go through these because you didn't get digestive system, so you got to make sure you know what the rectum is, right? And the anus, right? Because we really didn't go over that. And we didn't dissect the pig or anything. And this is the breast. I don't think I'm going to ask too many questions about the breast. I could show you the pictures of what you need to see. I think the next page has it. Like right here, you have the nipple, which has a duct in it called the lactiferous duct, and that's down here. You can see that. Lacto means milk. So the duct goes through the nipple, and the nipple is surrounded by a pigmented area called the areola. Then you have these, and these are mammary glands, right? These are mammary glands, which are modified sweat glands, right? Targeted, if you remember, by prolactin from the anterior pituitary to help with the production of the milk and also the maturation of the glands, the mammary glands. So the mammary glands have lobes. Then within the lobes, you have lobules. And within the lobules, you have cells called, believe it or not, they're called alveoli. So you can see how and there's ligaments that suspend the breast tissue, which is a lot of adipose tissue along the breasts. So the breasts are suspended, connected to remember the pectoralis major muscle, right? Which is the muscle of the chest, suspended with what are called suspensory ligaments. So the suspensory ligaments, and here's the muscles too, the intercostals too, and the minor, but it's mostly pec major. Right? You see the ribs on the lateral section here in the intercostal spaces. So go through this. Again, this is not on your list, but again, this will help you with lecture as well. So this is the male reproductive system. Remember the testes, right, on both sides. And these are the seminiferous tubules, and they're in lobules too. And this is the epididymis. This is your vas deferens on both sides, vas deferens. Here's your prostate gland. This is your seminal vesicles here. And these little glands right here are your bulbourethral glands. So you'll see, again, I'm going to use the real anatomy more likely. More likely, right? And there's a duct right here. The duct that's also going to the top of the prostate, and this is coming from the vas deferens at the point of the seminal vesicles, is the ejaculatory duct. So that happens right between the seminal vesicles and the prostate. And all this is urethra. Like this is the prostatic urethra. This is the membranous urethra. And this urethra right here is called the spongy urethra or penile urethra. And then you have the root of the penis, the shaft of the penis, and the head of the penis is called glans penis. And the opening is called the urethral orifice. And the prepuce is the foreskin. That's there sometimes cut away through circumcision. So now you can see right away, this is the male reproductive system because the sex organs and the gonads are actually outside the body, and that's for temperature. All right. So you see everything on the lateral view. This is the pubic symphysis right here. Okay, and again, there's only two openings, the anus and the urethra, as opposed to the female that has three. So this is kind of similar to the models, what you're seeing here. But again, you're going to practice in APR. And we're not doing meiosis and mitosis in the lab because we already did mitosis. So we'll leave that for lecture at this point. Okay.